CareDx is a transplantation-focused company, and we are dedicated to developing the best products to improve outcomes for transplant patients. Better matching and better surveillance equals better outcomes. Today we are going to talk about AlloSeq HCT, CareDx's kit-based solution to evaluate genetic chimerism in biological samples from recipients of hematopoietic stem cell transplantation. The assay input is genomic DNA extracted from whole blood or bone marrow samples, as well as from cell types or fractions derived from the primary biological samples. In this video, we are going to show you how easy it is to use and implement this assay in your lab. The instructions for use document will also showcase the detailed procedures of what we are going to describe here. Please read the instructions for use, also known as IFU document, to understand the limitations and contraindications, as well as other considerations before using AlloSeq HCT in your lab. Okay, so let's start. The sample set we will use contain pure gDNA samples from donor only and recipient only, which ideally should be obtained before transplantation. Processing these samples is important for the data analysis process after sequencing. The sample set also contains a number of post-transplant samples, which are a mix of recipient and donor gDNA, as well as a positive and negative control, which are optional. These controls are not provided in the kit, and water is used as a negative control. All gDNA samples that we will use in the assay are quantified with a fluorometric quantification method, such as qubit or picogreen, according to the manufacturer's instructions. Since we recommend the input of 10 nanogram of DNA per sample with the AlloSeq HCT assay, all samples have been previously normalized to 10 nanograms in 16 microliters volume using RNase DNase-free water in a final concentration of 0.625 nanograms per microliter. The first step in the protocol for library preparation is setting up a multiplex PCR. The following reagents are needed for PCR setup: AlloSeq HCT PCR mix, AlloSeq HCT PCR enzyme, AlloSeq HCT SNP primer pool, AlloSeq HCT index primers that include forward I5 and reverse I7. Since these reagents within the kit are stored at -15 to -25 degrees Celsius, first thaw reagents at room temperature, and then place on ice. Keep reagents on ice during setup. The PCR program should be set up for a reaction volume of 40 microliters. It requires specific ramp rate settings for the first 8 annealing at 57 degrees Celsius and elongation at 72 degrees steps. The ramp rate equivalents for different thermocyclers and PCR cycling protocols are provided in the tables within the instruction for use document. Let's get into the process now. In a PCR plate on ice, add 4 microliters of the forward index primer I5 to the sample wells. Keep track of which primers are being added to each well of the plate. This information is very important for preparing the sample sheet for sequencing later. Add 4 microliters of the reverse index primers I7 to the sample wells. Add 16 microliters of your prepared DNA to each sample well. Now let's focus on the master mix for PCR. Prepare a master mix in a 1.5 ml Eppendorf tube for your samples, using the table mentioned in the instructions for use. I have already prepared this using the table here. Vortex this master mix for 5 seconds to mix, and centrifuge briefly. Add 16 microliters of the prepared master mix to each sample well. Apply microseal B and immediately vortex the plate for 5 seconds to mix. Immediately centrifuge the plate at 1000 times G for 30 seconds. Check that no bubbles are present. Immediately place the plate on the thermocycler in a post-amplification area and run the PCR cycling protocol. The PCR cycling protocol takes approximately 1 hour. What happens during this reaction? Let me show you how this assay works. At its core, the AlloSeq HCT assay is a targeted next-generation sequencing assay that utilizes differences in single nucleotide polymorphism loci, also known as SNPs, to measure the amount of recipient and donor-derived %gDNA present in post-transplant samples. After a few rounds of specific primer amplification, additional primers present in the reaction containing index and adapter sequences anneal to the target-specific product, generating sequence-ready amplicons. That is, all 202 amplicons in the same well will contain a single set of sample-specific dual barcodes and Illumina-compatible adapters. The kit comes with a total of 4 I5 forward and 6 I7 reverse index primers that can be used up to 24 unique combinations. Therefore, 24 gDNA samples can be prepared in parallel for a single sequencing library. And that's how the assay works. Coming back to our protocol, now that our PCR process is complete, we pool our libraries, clean them up, and quantify before sequencing. For PCR product cleanup, you will need AlloSeq HCT purification beads and AlloSeq HCT resuspension buffer. Before you begin, make sure that you have removed the purification beads from 2 to 8 degrees Celsius storage and equilibrate to room temperature for at least 30 minutes. Also, remove resuspension buffer and PCR plate from 2 to 8 degrees Celsius and equilibrate to room temperature. Centrifuge at 1000 times G for 30 seconds. Prepare 80% ethanol, 3 mL total. Now let's mix the AlloSeq HCT purification beads and the PCR product. First, you need to determine the volume of each sample to pool into a final volume of 120 microliters, as in the examples in the table as shown in the IFU. For today's video, we are working with 2 samples. However, if we were working with 10 samples, we will pool 12 microliters from each library into a single 1.5 ml Eppendorf tube. Add 100 microliters of AlloSeq HCT purification beads to the tube of pooled samples. Vortex the tube for 5 seconds on full speed and incubate tube for 5 minutes at room temperature. Place the tube on a Dynamag 2 stand and allow beads to pellet for 5 minutes or until the liquid is clear. Keep the tube on the magnetic stand and remove all supernatant. Wash the beads twice with 80% ethanol, making sure not to lose any beads during the process. Remove the tube from the magnetic stand and add 35 microliters of the AlloSeq HCT resuspension buffer. Vortex the tube for 5 seconds on full speed and incubate tube for 5 minutes at room temperature. Place the tube on Dynamag 2 stand and allow beads to pellet for 5 minutes or until the liquid is clear. Without touching or disturbing the beads, transfer 32 microliters of supernatant using a smaller displacement pipette to a new 1.5 ml Eppendorf tube. Your libraries are now ready to be quantified and diluted before sequencing. Quantify it as per your usual methods. Dilute and spike in 1% Phi-X if required, and it's ready for sequencing. That's it for the hands-on protocol. After around 17 hours, the sequencing run will be completed, and the FASTQ files generated along with the sample sheet can be directly used for data analysis using the AlloSeq HCT software. Please refer to the AlloSeq HCT assay and software instructions for use documents to learn in detail about this protocol and the data analysis steps. But as you saw, it is easy to use and adapt in your own labs. If you have any questions, please reach out to your local CareDx representative. Thank you for your time.